as Tanya mentioned, I'm going to talk to you today about using EEG as a direct non-invasive measure of cortical target engagement in the clinical trial setting, particularly. So I didn't foresee Tanya giving me such a generous introduction, so I have very briefly, just to introduce who I am, added a slide. So as Tanya mentioned, I'm an assistant professor in physiology, and my research focuses largely around neurophysiology and how we can better measure changes in brain function using things like EEG and non-invasive brain stimulation. And I've been using EEG for just over 10 years now. And I've published a lot of work out of our lab and also some various reviews around how we can apply EEG to better detecting disease and then measuring the effects of therapies or disease progression on the central nervous system. So I guess before we get into EEG, often when I think about why we look at EEG, I think about what we're trying to achieve in a clinical trial in terms of measuring effects of the drug on the brain or the central nervous system. So, I mean, the main thing, of course, most fundamentally we want is safety. So what we're measuring and how we're measuring it needs to be done in a way that minimizes any risk to the participants. And of course, we also don't want to, be introducing any unnecessary procedural adverse events that might muddy our understanding of our drug-related adverse events. And then, of course, on top of safety, we have tolerability. So especially when we're doing trials in populations with different symptoms, experiencing different diseases, the tolerability that you might associate with a healthy population might be even more restricted in your group. And of course, we want to maximize the sample of the population that we can recruit to our trial. So we don't want to have to be excluding people because, for example, they aren't able to tolerate a certain procedure. Particularly, this is important where you have symptoms like impaired breathing, or, for example, if you're doing a trial in children. On top of that, once you have your safe and tolerable measure, it needs to be accessible. So is there the technology you need to record that measure of brain activity or brain changes? Is that physically in the setting where you want to do the clinical trial, or can you access a setting where that technology is available? Or can you bring the technology and the hardware and the software you need into the setting where you plan to do the trial? And is that cost effective, particularly when you have to do one measure in a very large sample or multiple measures, even in a small sample? The costs can really add up for some of the ways that we look at measuring central nervous system function. And then finally, flexibility is so important in a clinical trial. I mean, the day is so busy, you're recording lots of measures, you're on a very strict timeline, especially if you're looking at pharmacokinetics. So ideally, you want something that can be easily integrated into your desired timeline, that's time efficient, and ideally can be implemented in different areas, or potentially can be recorded in multiple people at once if you have multiple participants in on the same day. So looking at what we tend to use in clinical trials, I guess the cornerstone of measuring central nervous system effects in clinical trials is largely around measuring changes in symptoms, so functional assessments, cognitive assessments, motor assessments, looking at can we see any change in the symptoms that we're trying to alleviate. And of course, this will always be really important, but it's very hard to detect these changes specifically or particularly when you're on a very short time frame. So expecting that your drug is going to substantially alleviate symptoms in a matter of hours or days is possibly unrealistic. Also, these measures tend to be relatively insensitive. So detecting a clinically meaningful change can often be difficult, even if the drug is genuinely engaging the target or having beneficial effects on the underpinning biology. There's been a big move towards investigating fluid biomarkers, I guess particularly in the field of neurodegeneration and other neurological conditions. There's been a massive growth and interest of neurofilaments. So these are proteins that are released from damaged neurons. And certainly they can be very useful in looking at therapeutic effects, but they do require ideally a lumbar puncture, which again has the potential to introduce adverse effects. It requires specialist personnel and access to facilities. But as again, many of these fluid biomarkers require specific samples, access to certain lab personnel. So they have their challenges, and often they're not specific to engagement of a particular target, which can be, again, a limitation. I guess when we're looking at changes in brain structure and function, what we often think of most immediately is magnetic resonance imaging or other things like PET imaging or CT. And these are great ways of looking at the changes in brain structure, particularly in MRI, or changes in proxy measures or indirect measures of brain function. So in the case of brain structure, this is a really important fundamental tool, but I guess expecting your drug to change the volume or the physical connectivity of the brain, especially on a short time frame, perhaps might be unrealistic. And then when we're looking at changes in function, unfortunately, things like PET imaging or fMRI do not directly measure changes in neuronal function. They instead rely on indirect measures such as glucose metabolism or blood oxygenation, which in and of themselves might be affected by your drug. So that can make it unclear as to what exactly your drug is doing. So this brings me to talk about EEG or electroencephalography. Now, I'm trying to keep this webinar fairly light on the kind of nitty-gritty science physiology. So I'm only going to do one slide on kind of the in-depths of this. But for those who are interested in what is EEG or what is it measuring? So with EEG, it's a non-invasive technique where you're placing recording electrodes on the scalp. And what they do is record directly the underlying neuronal activity. So the way they do that is to measure the activity of aligned groups of pyramidal neurons. So in your cortex, you have these neurons called pyramidal neurons. They're so called because they're the shape of the cell body. And the signals coming into these neurons at what we call the apical dendrites are the signal coming in through ion channels generates a localized area of positive or negative charge in the surrounding cerebrospinal fluid. So you might have a localized area of negative charge, for example, if you have positive ions flowing into your dendrites. That might occur, for example, with glutamatergic signaling. And then to counterbalance that, you will have the opposing charge in the cerebrospinal fluid further along, closer to, for example, the soma of the neurons. What that does, in summary, is generates these two areas of separated electrical charge. And the electrical charge that is under the sensor, particularly where those signals are coming in, is what is detected by the sensors that we have on the scalp. So in that way, we're directly measuring when the neurons are receiving signals, and this can be from all different types of neurotransmitters. What we see in the EEG is a negative or a positive deflection in this signal. One of the great things about this is we can see these changes in positive or negative deflections on a millisecond by millisecond basis. So really fine-grained temporal resolution. So what are the pros of using EEG? So as I mentioned, it's a direct measurement of neuronal activity, as opposed to using indirect measures such as things like blood flow or glucose metabolism. And that not only helps to kind of provide a more specific measure of what your drug is doing in terms of regulating neuronal activity, but it has a number of other benefits. So first of all, you're measuring brain function. And as I mentioned before, that can be attractive, especially when you're looking at things like a short-term or immediate acute drug effects, as well as longer term drug effects. So you could expect that once your drug is in the cerebrospinal fluid at sufficient concentration, that neuronal function could be changed in a matter of minutes. And then that can be a prolonged effect going on hours or days. So you could expect to see changes in a very short time frame, which can be nice, for example, if you're doing a pharmacokinetic study. As I mentioned as well, these signals have really fine time resolution, which is something that's lacking if you're looking at something like an fMRI blood oxygenation signal. So the signals we look at, for example, with fMRI change kind of over several seconds. So there's a delay in the blood flow change that corresponds with neuronal activity change. Whereas we know that, of course, neural signaling depends on the temporal characteristics of the signaling. So it depends on what timing of signal change is happening between the neurons. So having that really fine-grained time resolution is really important because it allows you not only to look at activity, but also specific brain rhythms and how your drug is affecting them. It's also non-invasive, which of course is very attractive. And unlike many other methods, there's not really any adverse effects associated with EEG. It's also very relatively low cost. So access to the equipment and running costs is orders of magnitude lower than doing things like magnetic resonance imaging. Additionally, EEG is very portable. So, we have wired systems that can be easily brought to clinical trial settings and set up there, but also with the development and improvement of more mobile systems with wireless amplifiers, such as the one I'm showing here. This is the Smarting X Pro system. You can even allow people to move around without being plugged into any sort of immobile device. So, for example, you could set up your participants in the morning of the trial with the EEG cap and leave it on fairly comfortably all day for continuous recording or numerous recording points. Even if you wanted to do some home-based monitoring, some follow-up, or some screening, that could also be done with these portable systems. And then overall, a great thing about EEG is that it's very flexible in terms of protocol. So depending on what you're trying to measure, the protocols can be customized so to maximize participant comfort and to fit in with the timeline of your day. Like one great example of this flexibility is that for EEG, your participants don't have to be lying down. They could be sitting up, they could be comfortable, they could in different environments. It could be recorded at the bedside, for example. So it has a great ability to integrate into a clinical trial. So I guess in my kind of 10 years or so of using EEG, I've come up against these perceived cons or kind of biases against using EEG, and I just want to address those. The first one that I used to, not so much anymore, but I used to come up against a lot was this idea of that EEG is very noisy. So I've just put an example of a raw EEG signal in the image on the right-hand side here. And I'll say two things. First of all types of metrics of the central nervous system are noisy. Every measure of a fluid biomarker or any MRI or fMRI all have noise in them. But what I will say is that a lot of people are more familiar with the classic old style of looking at EEG where the raw EEG signal is read. Whereas what we're largely talking about is quantitative EEG, where we will record the signal and we can very readily apply processing pipelines, which will help to disentangle any noise there is in the signal from nice, clear EEG signals. So that's not really a concern. And of course, alongside that, we can design and test protocols that minimize any kind of environmental noise and maximize the quality of your signal to begin with. The other kind of perceived limitation of EEG is that it has poor spatial resolution. Now, this often isn't even a concern for clinical trials because if you don't need to know exactly where in the brain your signal is coming from, it's not an issue. You simply place the electrode over where you expect to see your outcome measure or where you want to see your outcome measure, and that's all the information you need. If you are concerned about spatial resolution, so if you want something that's more specific to a brain structure, there is now readily implementable source analysis or source localization algorithms that we can use to attribute your signal back to more specific brain areas. So this is just a figure from some of our work where you can see the signal can go from being a plot across the scalp to an image of a brain where you can localize those signals to specific brain areas. So how can we use EEG, or what are the different ways we can implement EEG in clinical trials? So I'll start off with what you might be familiar with, which is resting or continuous EEG. So here I'm talking about EEG where the signal is being recorded while the person isn't completing a specific task or engaging in a specific function. So we can do this when the person is totally at rest, or it can be a continuous monitoring while they're going about other activities. And by applying a montage of electrodes across the head, we can look at activity across the cortex. We can also look at connectivity, so what we call functional connectivity between different areas. So looking at how different areas of the brain are interacting with each other. And depending on what you're trying to achieve, this can be a protocol that lasts a small number of minutes. For example, if you want to get an idea of the resting cortical activity and connectivity, that can be recorded within 2 to 3 minutes. Or alternatively, you may wish to get data across the entire day. So the cap can be left on and can be continuously recorded for hours, particularly where we can apply those portable devices with the wireless amplifiers, that will allow for repeated sampling or continuous sampling across the day. So we use this in trials for a variety of different reasons. With continuous EEG, this can often be used for monitoring. So this is useful for a number of reasons, both in screening and for continuous monitoring of drug effects thereafter. So particularly, this is useful for detecting clinically silent neurological events. So things like ictal events or seizures, which can be seen in specific sensors. This can be really useful for screening participants for those at risk, or it can also be useful for monitoring after the drug has been administered for any potential risks. It can be recorded just at rest, but you can also apply things like hyperventilation or photic stimulation protocols, which can help to really test if there's any risk of adverse events that you might not see through a clinical assessment. And I've just highlighted here on the right this plot just to show that even without source localization, by applying different densities of montages across the head, we can look at where over the brain or over the scalp these signals are coming from. On top of that, we can actually get much richer information from the EEG signal, even in the absence of any adverse events, by decomposing the signal into different oscillations. So here I just have a cartoon of an example of a raw EEG signal. And what we know about EEG signals is actually they're a composition of lots of different EEG signals. So when we're recording them at the scalp, we're actually seeing a combination of lots of different communication going on between different neurons. So we can actually apply analysis to decompose this signal into those different communications, which happen at different frequencies. So by a frequency, I mean number of peaks and troughs that are in a wave per second. And we know that these different frequencies of communication represent different neurons and different networks and different cortical processes. So if you're familiar with EEG, you'll know these names. So we refer to them in order of increasing frequency as delta, theta, alpha, beta, and gamma. And these different oscillations in different brain areas are associated with different cortical functions. And that allows us to get this really rich information from across the scalp and allows us to look at if your drug or how your drug is modulating an array of different brain activity, all from this single signal. So just to give you an example of where we implemented this, this is an example of one of the trials that the science behind has worked with, and this phase 1 first in human, where the science behind provided the protocol design for the resting EEG, as well as operational support to implement that. The 32-channel EEG hardware was provided so that the data could be acquired, and quality control procedures were also provided by the science behind. And we also provided good clinical practice compliant data management and blinded transfer of that data. And this was all integrated in with pharmacokinetic sampling and other clinical study procedures. So this trial really demonstrates, among our other work, how easily EEG can successfully be integrated into a trial to add in some very complex and rich neurophysiological insights alongside everything else that you want to implement in your trial. Now, I won't go into the specific measures that were used in this trial because of confidentiality to our client, but if you'd like to see some more case studies of what we do, you can see those on the Science Behind website. So on top of kind of this resting EEG or ongoing EEG, you can also, if you're interested in very specific networks or cognitive functions or brain areas, you can look at task-linked EEG. So this I think of as a engage and measure type of protocol. So by using a task, having somebody do a task while you record the EEG, you are challenging their cortex to activate certain networks required to produce different brain functions. And what we can do with the EEG is then measure how well those brain networks are working and how that relates to somebody's ability to do the task. In fact, there is a lot of evidence to indicate that even if somebody is able to do a task normally, or apparently normally based on their performance measures, you can still detect abnormalities in how their brain is functioning. So for example, even if somebody is able to do a task just as well as a control population, their brain may be compensating by using different networks in lieu of some less functional networks. And this is particularly powerful then if you want a sensitive measure of how your drug is not just improving task function, but is able to target the underpinning biology that's affected by the condition you're aiming to treat. These protocols are quite flexible in that tasks can be customized. You can add breaks in between parts of the task to suit the tolerability of your participants. And also, depending on what you're trying to achieve, tasks can be various degrees of difficulty. So we might have more active tasks where the participant has to perform different responses. Or for some cases, we can use passive tasks. So we deliver sensory stimuli, for example, to the participant, and the brain is engaged, but the participant doesn't have to actively respond, which can be a very nice thing to have, for example, if you're working with children or people with certain cognitive impairments. So what does this look like in terms of EEG? So take a simple example of a task where your participant is undergoing EEG and you're playing a series of tones through some headphones. This is a very common example of an EEG related task. And you want to test their attention. So you want to see if they're able to detect a target tone. So let's say these blue notes represent your non-target tones and your red note reflects the target tone. So because of EEG's excellent temporal resolution, we can mark down to the millisecond where those tones were delivered to the person relative to their brain activity. So we can mark that in the EEG signal. And then by averaging the brain responses every time they hear a target tone versus a non-target tone, this averaging will remove any task irrelevant brain activity and leave us with a signal that corresponds to what the brain is doing to detect the target versus the non-target. Then what we can do is subtract the signals to leave us behind a signal that illustrates the brain activity specifically to do with the brain's detection of that target. So what we'll see in what we call an event-related potential is peaks or a specific peak. For example, this purple peak that I have in the bottom right-hand corner. And these are often used in clinical trials as outcome measures. you can measure both the size of the peak or the amplitude, or the latency of the peak, which means how long it takes for the peak to appear. And these can give you really great insights into whether certain brain networks associated with your task are suitably activated in that person, or if there is a slowing of the networks required to perform that task. And then, of course, you can measure if your drug normalizes or reduces that change that is associated, for example, with the disease you're studying. Without getting too technical, I just wanted to highlight that I did mention about how the brain communicates through different rhythms or oscillations. So here we're looking at the overall brain response during a task, which we can measure. What you can also do is look at specific oscillations or specific frequencies or rhythms of brain activity. So, for example, here, what you could look at is you could break that signal down to look at all the different frequencies in the signal, and then look at, do certain frequencies of signal, which represent certain aspects of neural communication, do they become more potent or less potent as someone's doing the task? So in this picture here, you can see the blue areas represent where an oscillation has become less potent while someone's listening to the tone, and the yellow represents where an oscillation becomes more potent. And much like I said before, you could look at this change in ongoing brain rhythms to the target and to the non-target, and then you could look at the difference. And what that will leave you with is a measure of the brain oscillation changes that are associated with performing a specific task or a specific function that you're interested in targeting with your drug. And then, of course, what you can do is measure this before and after, or in placebo versus controls, to see if your drug is changing this task-associated brain activity. So again, we've implemented these in various clinical trials. One example I'll give you is from a phase 1b study we did of an ion channel modulator in an epilepsy trial. So again, here are the science behind designed and customized the specific task and generated the software required to deliver that task and link it to the EEG. And then we provided on-site and remote training, as well as the 32-channel EEG hardware to implement that in a clinical trial site abroad. We also provided quality control of the signals, so between each participant, to make sure that signals were being collected to an adequate quality. And we also managed compliant data management and data transfer alongside subsequent EEG signal analysis. So we are able to provide customized signal analysis to extract, for example, the peak that you're looking at or the oscillation change you're looking at. And again, this was integrated with pharmacokinetic sampling and many other outcome measures to ensure a fluid delivery of the clinical trial. So again, demonstrating that you can get this really temporally precise monitoring of networks, in this case, particularly specific networks, for example, that you're targeting or that your disease affects, and see how that changes with your drug delivery. So overall, the science behind can really help you implement EEG in your trial from start to finish, or it can be we can tailor our involvement to suit your needs. So we can provide any or a combination of the following. So as I mentioned, we can generate tailored protocol designs. We can also test those protocols to ensure that they can be fluidly delivered in the time frame that you need. We can also provide directly the personnel to record the data, or we can provide training of your local personnel to implement these protocols. We also can put together packages of customized equipment to suit single site or multi-site studies, so that you can access the hardware and software you need. We can also work with your hardware if you already have existing EEG equipment on site. And then we can provide a suite of different analysis and reporting options. So we do everything from fast turnover quality control up to EEG signal analysis, statistical analysis, and reporting if and as needed. So that's quite a quick oversight of some of the work that the science behind does to implement EEG in clinical trials. So thank you very much for your time, and I'd be happy to take any questions if you have any.